I have the pleasure to introduce today's speaker, Abby DeCruz, who is Head of Strategy and Operations at Probably Genetic. Probably Genetic develops technology to find undiagnosed patients online and helps them get tested. Abby's an expert in genomics, holds a PhD from the University of Oxford, and specializes in rare genetic diseases. Probably Genetic develops technology to find undiagnosed patients online and helps them get tested. Before joining Probably Genetic, Abby was a senior consultant at Trinity Life Sciences. Welcome, Abby. Tell us a little bit more about Probably Genetic and what drove you to join the company.
Hi, everyone. Thanks very much for having me. Really excited to be here and with the team at the SRF as well. So thanks, Corey, for the introduction. And it's a great question because I've been with Probably Genetic coming up to, I think, 4 or 5 months now. And as you mentioned, my background after my PhD, which kind of was oriented around clinical genetics, very much in a lab research focused position. After that stint, I joined Trinity Life Sciences, where I really had an exposure to a whole variety of therapies and also disease areas. But I developed a sort of a focus on rare diseases, orphan conditions, and then also therapies that were being developed across those indications. So through that position, I think got a really broad exposure to some of the challenges that drug development faces within those settings. And I think through Probably Genetic, I've been able to really zero in on the most, I think, pressing challenges that occur in this space, and some of the most kind of incredible and innovative work that's going on to address those challenges. Because, the mission, as you just mentioned, Corey, at Probably Genetic, we have a very aspirational mission to diagnose 200 million genetic conditions, to diagnose 200 million patients with the rare genetic conditions globally. And that's obviously a pretty impressive target, but we're doing that through really innovative ways that I think are really exciting. And we're kind of looking to leverage some very sort of cutting edge and newly developed ideas that merge technology, in addition to all of the ridiculously advanced research that's currently going on in the genetic field. So looking to merge a couple of my areas of interest and tackling challenges that I've come to understand are really complex and obviously, incredibly worthwhile. So we managed to tick quite a few boxes at Probably Genetic, and the work is incredibly rewarding, as I think we're going to get into today.
Wonderful. Look forward to it.
Perfect. Well, I think everyone can see the screen that we've put up today. So we've got a short presentation where we can go into really some, background on what it was that we put together with the SRF, and what the kind of objectives were for this partnership, and excitingly, what's kind of come out of that program, and looking ahead to the future of next steps as well. So to kick it off at a very high level, our objective was to figure out whether or not we could identify SYNGAP1, your sort of target condition of interest, using machine learning effectively. So what we've broken this down into in the next slide, if it will move, there we go. To pursue that objective of developing what is effectively a symptom checker that we could use to identify SYNGAP1 patients, we broke that down into essentially a 5-step process. So the first step was to develop a prototype, which essentially involved the Probably Genetic team working really closely with the SRF as well, to leverage a lot of research that's out there currently, in addition to patient feedback, family feedback, carer feedback, to develop a prototype symptom checker. So what we mean by that, really at the very initial stages, was just effectively a list of symptoms that would be associated with the target condition, so SYNGAP1, but also a list of symptoms that would be associated with differential diagnoses that patients experience in their diagnostic odyssey, and then also just kind of peripheral symptoms that might crop up as well. So we're really trying to capture a comprehensive list of symptoms that our patients might encounter within their journey, and feed them into what we developed via our platform was a really user-friendly symptom checker that patients could go in and, for example, use more patient-friendly terms for some of these phenotypic symptoms that might crop up. So we initially developed that prototype, and then what we did, again leveraging the SRF community, was that we put that out for feedback effectively. We put that prototype out to say, are these the symptoms that you're seeing? Is this comprehensive? Is this accurate? Is there anything that we've missed, really importantly? Are there any terms that maybe are surprising to you, or that would help you to use to identify what's going on with you in a way that would enable you to input this into a data capture form. So we honed that prototype in the feedback step. And once we were pretty confident that we had a really comprehensive and accurate symptom checker, we then set out to collect training data effectively. So our target here was on 100 patients. So what we mean by collecting training data is essentially just getting people to fill out the symptom checker. So sending this link out that was live on, definitely within the SRF community, lives on your website as well. We got an enormous response, but effectively we just wanted to get that link out to as many people as possible so that we can collect training data on patients within the community to see how those responses look when we submit, this huge list of symptoms to them, what's coming up, right? So what are the patients within this community? How are they characterizing their symptoms? What's what we call a phenotypic profile for this community? So that was kind of the training data step. And like I mentioned, we'll go into data a little bit later, but our target there was for 100 patients for a really good comprehensive snapshot. So once we've developed the symptom checker, we've received the feedback on it, we've basically unleashed it on the community. We're then training it. So the training step, it's encompassed in 3 words here, but it's a little bit more complex than that. This is where our machine learning technology, our sort of algorithm-driven technology comes into play. So what the symptom checker can do with our machine learning algorithms is it will ideally get really good at distinguishing patients that have the target condition to SYNGAP1 from other patients or patients that don't have SYNGAP1. So we would know that in the, within the community, we know patients that have previously received a diagnosis of SYNGAP1. So the goal there, when we say training the symptom checker, is that we would get it to a point where it becomes really good at identifying when someone who's submitted their phenotypic profile to the symptom checker has or a high likelihood or does have SYNGAP1 as the target condition. So that's that 4th step. And then finally, the kind of the why or the so what of this whole exercise is to basically provide the SRF with an incredibly rich source of, information, really useful information for their community. And I think that the 5th step is obviously the really exciting one, definitely for the SRF, but also for us, because, that's the real value add, I think, and that really enables us to zoom out a little bit on the why. Because, as we know, and as certainly everyone at the SRF knows, within the rare disease community, the more patients that you can find and the more you can draw attention to how many people are either living with your target condition or, trying to find answers in the space within which your kind of condition exists, the more people you can identify in that space, the more attention you can drive onto that disease, the more potential R&D effort you can drive, and the whole kind of, biotech and research cycle can have a bit more energy put into it. So that was a little bit of an overview of the initial 5 steps. Let me pause, Corey, to see if we've had any feedback or questions or if I can keep going.
Let's see. I don't see any questions as of yet.
Great. I must be doing a spectacular job, or they're all just holding up. So I'll keep going. So this is basically the overview where we've broken down into steps how we were kind of addressing this overall objective to develop a symptom checker to identify SYNGAP1 patients. So let's get into how this actually fared. So like I mentioned just before, the initial objective was that, in that training step where we were going to collect submissions, we were targeting 100 as a really healthy data set. What we achieved at the end of our initial pilot was 603 unique submissions, and that was right across the globe, certainly right across the United States. And you can see from on the left, the growth tracker in terms of number of symptom checkers. This program had just phenomenal growth, which was powered by the enormous level of motivation and engagement driven within the SRF for people to submit their symptoms and create those symptom checker submissions. So we had we witnessed rapid growth of the program, and the geographic spread as well is a really powerful, I think, example of just how much appetite there is, and just how just the need, I guess, the high level of need for people out there who are looking for answers, people out there who are looking for remote testing programs and patient-initiated solutions. So we love this slide. It really illustrates the scale of, what we're doing, and certainly illustrates how motivated the SRF community is in particular to, click on the link basically and distribute the link to patients and their families who are looking for answers. So what we're at now is 3,287 unique submissions, and we're seeing that continue to grow and seeing that to continue to come in from around the world. So massively exceeded our growth expectations in terms of program submissions. So the next piece is just what those submissions, or what that kind of snapshot of the community looks like. So the people that are submitting to this program, again, this is for the current data. So I think by the end of the pilot, we had about 80 SYNGAP1 patients submit to the program. We're currently at 100 SYNGAP1 patients submitting to the program, and 20 different diagnoses as well that are submitting phenotypes to the program, and contributing to the richness of that data set, and contributing to the richness of information that we have of patients that are within the SRF community. So this is a really powerful graphic as well, just because it illustrates just the variety of differential diagnoses, and also just the variety of conditions that are present within this community, which again, on this graph, we're showing some incredibly rare disorders. And again, it shows, what we think kind of underlines the diagnostic odyssey that a lot of patients go through. And again, provides a lot of really, really useful information for the SRF and similar organizations that are really looking to shed light on some of these conditions. So some pretty extraordinary numbers in here. So on the next slide, what we're showing here, what we think is really powerful is, in terms of achieving that objective of a symptom checker that can identify SYNGAP1 patients. What we found through the program is that SYNGAP1 has that distinct phenotypic profile as compared to patients that don't have SYNGAP1. So those are the patients that haven't submitted, a diagnosis of SYNGAP. So what we're seeing on the left here is essentially a ranked snapshot of the symptoms of everyone that self-identified as having SYNGAP1. So those are 84 patients that we use there. And you can see that the global developmental delay, the intellectual disability, motor delay are the top symptoms that are incredibly frequent within patients that have the SYNGAP1 diagnosis. Whereas on the right-hand side, you can see that patients that don't have that SYNGAP1, their phenotypic profile often looks very, very different to those SYNGAP1 patients. So this is really just underlining the power, sort of addressing the question that we had around, can our technology, can a very comprehensive symptom checker and our machine learning algorithms, can that, with a high degree of likelihood, identify a SYNGAP1 patient from a non-SYNGAP1 patient? And what we found is that, look, the snapshot looks pretty good. This is really promising. This is what we like to see. Again, we love this slide because it's just sort of illustrating the power of this program, because it is a patient-initiated program. So we're just sort of putting this out there in the community, and these kind of stark results is really kind of underlining the power of the program in terms of predictiveness. So we're just going to dive a little bit into some of the, I guess, the science, or the step 4, if you remember, of the training for our tech, because we think that this is kind of really powerful, and it's sort of the nuts and bolts of the slide that you saw previously. So this is just a little bit on what our model actually does. And what it does is that it will look at all of the users that are the SYNGAP1 submissions, all of those profiles, and it looks at the symptoms that those patients submitted, and it'll rank those symptoms by importance. So what that means is essentially, if there's a symptom that the vast majority of patients with SYNGAP have, that'll be a really important symptom for the model. It'll prioritize that symptom and kind of flag it as something that is seen very often with SYNGAP1 patients. And it'll also mean that, the importance of that symptom will increase relative to non-SYNGAP1 patients. So This is basically just a quick look behind the scenes. And again, really important information for communities like the SRF, where they're looking to really sort of have an indication of the symptoms, not only that their patients are experiencing, but also the symptoms that really distinguish or differentiate their target condition, and can be used to sort of identify patients that have a high likelihood of having SYNGAP1 in this instance. So this is what we refer to as feature importances, but it's essentially just allocating an importance to the submitted symptoms. So what we've called out here is just a couple of examples where, what this enables us to do. So for example, it will enable us to identify, for example, a symptom that might be common for SYNGAP1 patients, but rarer for non-SYNGAP1 patients. So what that'll enable us to do is to flag that symptoms as an important one, because it can potentially distinguish between someone that has SYNGAP1 and someone that doesn't, because it's effectively telling us, okay, this is a common symptom in SYNGAP1 patients, but it isn't in confirmed non-SYNGAP1 submissions. This is really powerful information when you're looking to, go into that sort of characterization of a patient community and flag those symptoms that are really important for your target disease. So the so what of everything that I've kind of just said in terms of, the things that our model is doing behind the scenes to identify important symptoms and to be able to distinguish SYNGAP1 users is basically shown on this graph. So the key message here is that through all of those mechanisms that our machine learning model uses, we found that we can use it to identify 97% of SYNGAP1 testers. So what that means is, if you had 100 SYNGAP1 patients that submitted to the program, the machine learning model that we trained just by itself would be able to identify 97 of those SYNGAP1 patients. So a really, really effective predictive rate, basically. What we did there was, what this graph is showing is that we identified a threshold that we used to basically score submissions to say this SYNGAP1 submission, is kind of over the threshold and therefore has a high likelihood of having SYNGAP1 or would therefore be referred to testing or something like that. So the slide here is basically showing that the ML model has a really high ability to identify SYNGAP1 testers, which is really good news for us based on all of the work that we did with the SRF, training the symptom checker, and then having it be able to identify SYNGAP1 patients. So this is kind of the outcome slide, and this is accurate for the end of the pilot. So I won't get too much into this, but the key message here is that with the ML model that we trained working with the SRF, we had 81 SYNGAP submissions that would be offered testing. And really importantly, we had 28 non-SYNGAP1 submissions that we would also offer testing. So those are patients that we've that have submitted to not having a SYNGAP1 test, not having a SYNGAP1 diagnosis, but that the program would suggest to offer testing to, which is really interesting and valuable for us. And that's only at the end of the pilot. So, we've collected a whole bunch more submissions since then, and we would anticipate that number will go up. So this is exciting on two levels. The first is, that we're identifying patients that, the model is kind of accurately identifying patients that would be offered testing because they, look like SYNGAP1 patients. But really excitingly is that it's identifying patients that haven't currently got a diagnosis of SYNGAP1 that we think should be or would be offered testing, which is great. So again, really valuable for organizations like the SRF, who are not only interested in identifying patients, in learning more about their community, but also in identifying patients. So it was really good news for us, certainly, but also this kind of data is really exciting because it hits on both of those kind of key objectives for patient advocacy organizations, and certainly for ones as engaged as the SRF. So this is kind of diving in a little bit to to the previous phenotypic profile slide that I showed, where if you kind of double click, so on the previous slide, we're kind of zooming in now on the 28 and the 81. So of the 28 and 81, so over here on the left-hand side, where we know these are patients that have SYNGAP1, that's their phenotypic profile. And this is just kind of illustrating that of those 28 patients that don't have a diagnosis of SYNGAP1, that the program has sort of flagged as, should or would offer testing for SYNGAP1, their phenotypic profile looks much more similar to patients that do have SYNGAP1. So this is basically, the foundation upon which our program suggests that we would offer, that kind of would flag these patients, that the SRF can, connect with these patients to link them with additional information and resources. This is the basis for that, where we're saying, hey, these patients have a phenotypic profile that we think is pretty similar to a SYNGAP1 patient, and therefore it might be helpful that we, connect them to resources and ideally additional testing. So this is a really sort of exciting and obviously useful output for programs for foundations like the SRF, but also is kind of the key reason behind programs that we run like this, where we can hopefully use our technology to identify patients that haven't yet been given a diagnosis, but that could certainly be really good candidates for additional information and potentially linking and connecting to testing resources as well. So, in summary, essentially, we wanted to show this slide as kind of a snapshot of at the end of the pilot. So at the end of our pilot that we put together with the SRF, we had that objective that I spoke about earlier to develop a symptom checker for SYNGAP1, and we had broken that down into 5 steps. So the great news was that we met or exceeded all of those steps. We developed the prototype and received feedback with a huge amount of help with by the SRF and their community. We had that training data, huge overshoot, 603 patients ended up submitting, so a really powerful training set that we were able to run the symptom checker on and developed what I've hopefully just shown you to be a really accurate and highly predictive symptom checker that's really powerful for SYNGAP1. And then we were able to provide a huge amount of really exciting data to the SRF as well, that has hopefully really empowered them and provided them with an additional very rich resource to continually sort of drive engagement and effectiveness within their community. And we had sort of put in as kind of aspirational bonuses. I remember at the end of our sort of pilot report with the SRF that we had 2 additional points that we were really excited to get going on, which was firstly to build a tool for the SRF to engage your community and your patients, where we could put all of this data and all of these abilities together into a really user-friendly portal. And then the second piece, which is kind of the most important bit, was to launch a free testing program so that we can basically connect these patients that we're flagging as potential candidates for SYNGAP1. We can connect them to that potential answer in the form of free testing. So this is what we kind of showed at the end of the pilot that we put together with the SRF, and we were really delighted to have met all of the project objectives. But the exciting update, and I don't even know that the SRF knows this at the moment, but the excited update that we have to share today is that we actually have just signed a partner for the free testing program. And we've just signed with Marcy Therapeutics, who are going to basically work with us to essentially be able to offer, potentially offer free testing to any of the SRF submissions that are eligible. So we have that capability, kind of the back end to this in process, which is really, really exciting and kind of connects up the, the finding with the additional sort of diagnosis piece as well. So that's a piece that we're really excited to get working on, and was our sort of eventual goal. So that's a really exciting next step that we have to come.
Wonderful. I know everybody is really exciting about the free testing program.
So are we, yeah.
What else? Let's see. We've got a couple questions.
Mm-hmm.
Let's see if I can get them pulled up. Let's see.
Before you jump into that, Corey, can you? I have a question, Abby. So on bullet 3 there, you say collect training data on 100, 600, 3,200. So that's amazing, right? So we've got 3,000 plus people have taken this. What's the current number for patients who are testing probably SYNGAP? Because I think earlier on you had a slide around 28, but I feel like that number's gone up as well. Do you have that at your fingertips? Or
that number has gone up. I don't have it at my fingertips currently. I believe it was over 100. That was less than 2 weeks ago, but I'd have to double check that. But it's gone up significantly.
That's awesome. That's awesome.
Yeah, which is super exciting.
And the Mozzie thing, like the ink is drying on the paper, right? So we don't even have a press release on that yet.
That's exactly right. Yeah, the ink is drying as we speak.
We should work on that press release.
Oh yeah, we absolutely should.
I am so excited. This is great. Okay, Corey, that was my question. At your show.
Let's see. On the slide with the 20 different diagnoses listed, to confirm, out of the 3,000 submissions, just have 100 have SYNGAP diagnosis. So we kind of answered that just now. Well, the 100 was the patient population. That was us that filled out where it says SYNGAP1. So that was actually our people in this list, correct?
Yeah, so actually this is the snapshot of all of those submissions. So this would be the top diagnoses. So these are the patient reported diagnoses. So, you know, when you fill out the symptom checker, you can input any diagnoses that you've received clinically. So of those, so the question is correct. That's right. Out of the 3,200 odd submissions, 100 have self-identified as having SYNGAP, and then you can see the prevalence of the other sort of top diagnoses, so to speak, noted on the slide. Yeah. So that's right.
So we found, in addition to the 100 that we already knew about, we found the additional 100 patients that fit that profile out of the 3,200. All right. And says next, this is from Ed, and the slide showing the symptoms of SYNGAP1 versus non-SYNGAP1. I assume that all other disorders are lumped into the non-SYNGAP1 chart. If so, have you looked into whether there are any individual disorders within that group that mirror SYNGAP1 more closely than aggregate of all non-SYNGAP1? So I guess
I think I understand this in terms of
is there
differential.
one particular phenotype that fits SYNGAP1 out of this bunch more so than the rest of them?
Yeah, so that's a really interesting question. So the short answer is no, in that whether or not we've run an analysis that stratifies non-SYNGAP1 submissions according to likeliness or likeness to the SYNGAP1 phenotypic profile. And the reason that we haven't done that is that it would be quite intensive, because you'd have to sort of stratify. You probably saw that list of differential diagnoses, and then do a comparison, which would involve retraining the model. Whereas the approach here is to just sort of use the knowns. So what I mean by that is, you know, the knowns for this training, for this program, and the way in which we set up the training is that we had our one known be the SYNGAP, and then had, you know, the sort of differential diagnoses come in. So if we wanted to run something like what you're proposing, we'd have to make sure that that initial step where we, you know, did the really comprehensive set of symptoms for each of for SYNGAP1, and then the comprehensive set for all of SYNGAP1's differential disorders, that we would do that set for every diagnoses that we'd want to compare to SYNGAP, which, you know, obviously would be quite a few. So the short answer to your question is no. We currently just have the SYNGAP1 known comparison to the non-SYNGAP1 submissions.
He said, did any of them previously have a different diagnosis?
Yes, several. So I assume you mean the SYNGAP1. So that's another really, really rich you know, source or resource, both for us and for the SRF. So when patients submit to the symptom checker, they can submit, you know, obviously a really rich amount of detail on their symptoms, but they also submit information on things like onset, on things like severity. And so what we can map from that for the target condition is a very, very detailed, comprehensive, almost, we like to call it the diagnostic odyssey, right? Where we can kind of say, all right, the majority of SYNGAP1 patients are experiencing an age of onset around about this time. The majority are getting a diagnosis around about this time, which, you know, allows you to calculate kind of the the average weight effectively for a diagnosis. All of those data points are, in many obvious ways, very useful, not only for the SRF, but for everyone in the Syngap community. But certainly it's because it's all patient initiated and patient reported, it's a really powerful way of getting a snapshot on what patients themselves and patient families and caregivers are experiencing, and kind of the ways in which they characterize their own experience, which I know that the SRF is really excited about, and we found to be a really powerful tool.
Yeah, I'll tell you, looking through the data that we received, it was very interesting to see how many people had already had some type of genetic testing in the past, but it
Mm-hmm.
has been, 10 years, 5 years, probably back when they weren't testing for SYNGAP.
Mm-hmm. Mm-hmm.
So we've gone back and made a recommendation to them to go back to the genetic company that did the testing and get them to rerun the port. It's free to them. And there might be something, even if it's not SYNGAP, something else might pop up that wasn't previously known at the time they had the original genetic testing done.
Yeah, that's exactly it, and that really underlines the power of the kind of, I guess, sister slide to this one, where we're showing the profiles of the patients that we're saying, hey, these didn't submit a SYNGAP1 diagnosis, but they really, the model's telling us that they really maybe have a high likelihood of either being a patient or being very close to, or having a phenotypic profile that would really benefit either from additional testing, maybe revisiting testing like you mentioned, Corey, or just additional resources and just a little bit more to learn about the specifics of the disease. So in any of regardless of which bucket you fall into in terms of patient and diagnosis and what your previous testing history was, which we also captured, by the way. So yeah, Corey, we were looking at that data in terms of the tests that patients have previously had. No matter what bucket you fall into, we're hoping that the program is producing a resource that's useful for all of those patients.
Yeah, because so many people don't realize you can go back and have it rerun. They don't realize the differences between the different types of genetic testing. Not everyone tests for everything. So it's important that they go back and really try to fight for that diagnosis, because so many people just don't know to ask the different questions.
That's exactly it. Yeah, and it really speaks to the SRFs, particularly good at this, but the role of patient advocacy orgs of that empowerment piece, right? So even if we're not saying, oh, you look a lot like a SYNGAP1 patient, that the power of that knowledge that you can go through, hey, you can ask your doctor to revisit this, you can ask your testing service to revisit this, or hey, look at this gene or this variant or whatever it might be. Those are the really kind of empowering pieces that this kind of information can hopefully provide a link to.
I also wanted to make note, we did create a Probably SYNGAP Facebook page. So anybody that got a high probability test result back from the survey, we made sure we've let them know, hey, we've got a Facebook page. It's a Probably SYNGAP. And you can kind of go in here and talk to other parents that have also tested highly probable, and talk about your stories and different things about your kids. And it's been really great for them to have a home too, because they've been so long without knowing, and just at the very least finding people that have similar phenotypes as their children has been so beneficial to them.
Yeah, I mean that was just such an incredible piece of this program. I know that in our we had weekly checkpoints as we were running the program, and certainly hearing from the parents, hearing the testimonials that came in. I mean, we had people who were using the program that had just been like elbows deep in this diagnostic odyssey for just years and years and years, and like any light that you can shed there is just so impactful and powerful. And so being able to hear all of that through the SRF, and seeing the impact that you guys are having, and that we can have any small hand in providing you with any tools or resources, that was definitely really powerful for us. And I think just exactly what you said, Corey, the impact of information, the impact of community was definitely underlined by this program. And yeah, I mean, that growth curve, like Mike said, that growth curve speaks for itself. The SRF community enormously engaged, and yeah, you guys are definitely a role model for all the other programs we're putting together.
We've been hanging out in the undiagnosed parent groups a lot, and
Yeah.
and it's funny, you can kind of hang out in those groups and see when they talk about their children that they seem to have very similar symptoms. And so we try to reach out and put the link in there to let them know, hey, here's a chance to maybe kind of narrow down your odyssey. So hopefully we'll continue to find some more people out of that. Sorry, my dog went nuts.
That's okay. I mean, that's actually a great segue, and I'm going to pounce at that opportunity, Corey, to basically just make that exact point. So, there's a reason why I've just flipped to this slide again. Obviously, we're incredibly excited. SRF is really excited that we've signed a partner to connect up these two pieces, the patient finding and then the actual testing or the diagnosis piece, which we're really excited about. But soon what that means in terms of next steps and what your community is going to see is that soon we're going to have the whole process connected up, and the people within your community are going to start to see the link pop up, and they're going to have that opportunity to be considered for the free genetic testing. And we can only implore you and everyone in the community to do exactly what you just mentioned, right? If there's any corner of the internet, of social media, of any kind of disease influences and whatnot that you interact with during your diagnostic odyssey, that you can get a link to or just share some information with about the program, that's so powerful, and that's exactly what we need, because all we're trying to do is all we can do is develop mechanisms, right, and develop tools. And it's really just the communities that we serve, so the communities like the SRF and patient communities, that are the power behind that. So you guys really power the mechanism, and it's only without the actual SRF community, we wouldn't have any of the results that I was just able to show today. And I think that the fact that all of our expectations are exceeded, and we've been able to produce this really data rich, really important, sort of powerful resource that's giving us so many insights on the community. That's all down to the community itself. It's we can develop all the ML tools in the world, but they've got to be used. So we're really excited to have this opportunity come up, and we're excited to see you guys put the link in every corner of the internet once again.
One thing I would like to you to speak on is the privacy. While we are in these these undiagnosed parents groups or epilepsy groups, there has been some hesitancy for people to give their information. Information is very critical nowadays, and people are much more hesitant to give out that data. Could you speak to a little bit to ease some of the people that may be more hesitant to fill out a survey because they're worried about that information being sent?
Yeah, absolutely. And it's a key point, and we really understand this. We've got a couple of, our team members who have rare genetic conditions, and so have, firsthand experience of, yes, you're on that diagnostic odyssey and you're really looking for answers, but also you have that wariness, right, about your data. So the data privacy has been centered in how we've developed the symptom checker and also all of our processes. So from end to end. So what that means is from the point at which a user even clicks on a link to the symptom checker or enters any information whatsoever, we're compliant with everything that you need to be compliant with. So the primary piece there is HIPAA. Every single aspect of our system. So not only our website where you input any information and the databases that we use, those are all HIPAA compliant, which protects your data and means that it's all completely adherent to the relevant laws that protect any personal identifying health information. So that is all very secure in terms of where any information is stored, if at all. And that's end to end. So that's not only when you input your information, it's also at any point, when your data is sent to our sequencing provider, to our report generator, to our analysis, it's all completely HIPAA compliant all the way through, it's a bit of a gray area whether that's actually required, but we've kind of gone through like ironclad and made sure that every single step in our process is compliant. So there's that piece where, on an institutional, on a legislative level, we're completely, almost extra compliant. But the second piece is, our responsibility. So we take this really seriously around how we use data that we would have. And there's two key pieces here. The first is that anything that is personally identifiable, anything that could be in any way linked back to you or anything like that, is completely, de-anonymized and also is protected by consent. So we'll never share any part of your data without your explicit consent. So nothing is assumed. The consent has to be explicit. And so that's the first piece around explicit consent of personal health data. And then the second piece is that any data that we do share, so for example, the data that I just showed everyone in this presentation, everything is anonymized in a very stringent way. So for example, any location information, so when you complete the symptom checker, that IP even is completely anonymized to zip3 level. So, the last 2 digits of any zip is taken out. So even those dots, if you zoomed in on that location map, all you would get is like a pretty sizable region, because that would be the zip3 level. So you wouldn't be able to see anything further than that. So that kind of thing as well, we take really seriously. So any kind of information that would be passed on to any prospective partner, even for example, would be completely de-anonymized, and again, would require that consent. So I think that those are, I could probably talk about that for a long time, but the two key pieces are basically that we take it really seriously, and that's driven by the patients that we have within our organization. And the second piece is that we're, extra compliant with all of the regulations that serve health data.
That's great. Thank you very much for that. All right. And then we did have one more question that came through from an anonymous attendee. She said, just to clarify, how many months were the 3,200 submissions over?
Yeah, that's a great point. I can actually jump to that. So it's from, I think about Feb. Yeah, there we go. So basically from early Feb to June. So what's that, about 4 months?
Yeah.
Is that right? Yeah. So it's honestly, it's astonishing growth. I think by the end of the pilot, like I mentioned, we had, I think, 600. So you can see the growth just from that period alone. We're seeing submissions continue to come in. So, we're still getting, I think, upwards of 50 a week. And even that is like a slight drop off. You can see that the rates kind of evened off a little bit in recent days. But that growth trend with that, the rate of growth is remarkable and definitely exceeded everything that we thought would happen. So, yeah, about 4 months to date.
Great. All right. Well, I think we are done with questions, unless anybody has a last minute one they want to pop in the chat. We appreciate it, Abby, and we look forward
it's been a pleasure.
finding out more and getting more data from you guys. It's amazing.
It's been great. Yeah, we're so excited for next steps. So yeah, can't wait for everything that's about to happen. And yeah, definitely echoing Mike's excitement. We're really raring to go on this.
Well, you have a wonderful day.
Thanks, everybody. You too. Thanks for having me.